Today I've been asked to talk about root knot nematodes and specifically some information about Guava root knot nematode, which is a new pest in Australia. All right, so yeah, I'll just give a bit of background on nematodes, move on to root knot nematodes, and then some information on Guava root knot, just a bit of background on that pest and some of the research we're currently doing and some we've just completed. A few tips on managing root knot nematodes. And then I'll introduce Ufa, who will be our New South Wales collaborator for the National Guava Root Knot Project. So nematodes, facts and figures. They're unsegmented roundworms. They're mostly microscopic, but can be up to 8.4 meters long. I think that that record-holding one is found inside a blue whale. They're mostly free living, but many of them are important parasites of insects, animals, and plants. They're found everywhere on Earth. They're ubiquitous. And as a fun fact, they're the most abundant multicellular animals on Earth. By numbers, 80% of all multicellular animals are nematodes. There are 60 billion nematodes for each human, so that'll make you feel insignificant. In the soil, there's a huge diversity of nematodes. Most of them don't affect plants. They feed on bacteria, fungi, combination of things or each other. And in fact, many of them are beneficial. We can use them as soil health indicators. They're reasonably easy to identify by their mouth parts and a few other features as to what they feed on, what type of nematodes they are, and they respond pretty quickly to changes in the soil. So they are good soil health indicators. And they can perform important services in the soil, like mineralizing nitrogen. So, for example, in a healthy soil, fungal and bacterial nematodes can excrete around 50 kilos of nitrogen per hectare per year. So that's not insignificant. Down the bottom there, you can see a photo. That's just a typical extract from a soil sample. There's always lots and lots of nematodes. As I said, most of them aren't plant parasites, but there is a huge diversity in the soil. Moving on to plant parasites, they're characterized by having that retractable spear or stylet, which they use to pierce plant cells for feeding, and they've got a muscular bulb in their esophagus to pump the plant cell contents into their gut. There's only about 25 widespread or important genera of plant parasites, and most of them are pretty small, less than a millimeter. They have different life cycle strategies. There are ectoparasites, for example, dagger nematodes, ring nematodes. They sort of feed on the exterior of roots. There are semi-endoparasites, which sort of embed themselves in the surface of roots. So reniform nematode, citrus nematode, examples of that strategy. And then there are the endoparasites. So there are sedentary endoparasites. So our root knot nematodes are sedentary endoparasites. Once they invade the root and start feeding, they don't move. They just stay in the same spot. And then there are migratory endoparasites like the lesion nematodes, burrowing nematodes, which move through the roots as they're feeding. Yeah, so root knot nematodes, there's over 100 species worldwide, and most of them have pretty wide host ranges. They're considered the most important nematode pests in horticulture. They obviously cause galling on the roots, and this leads to a lack of vigor and symptoms like yellowing and wilting. Because of that damage and feeding, they reduce yield. And root and tuber crops also suffer some direct damage from the feeding of nematodes. You get downgrades on the marketable product. They've got pretty short life cycles, so it can build up to large numbers in long crops. They prefer generally lighter sandy type soils, but also do pretty well in well-structured clay loams like ferrosols that we find in some of our excuse me, horticultural areas. There's only a few species in Australia, and most of them well established, have been here for a long time and are pretty widespread. So there's 3 warm climate species, Incognita, Javanica, and Arenaria, and 2 that are sort of called cool climate species, Meloidogone hapla and Phalanx. Those warm climate species are found, despite the name, are found pretty much all around Australia. And of course, we have the recent introduction of Meloidogone enterolovii, which is the Guava root knot nematode. So the life cycle of root knot nematodes, they start as eggs laid on the exterior of the root by the female nematode. She can lay 1,000 eggs. They have one molt inside the egg and then hatch as a second stage juvenile, which is the typical worm-like nematode. They migrate through water films in the soil in response to plant exudates, and then wound the root and enter the root before moving to a feeding site. So they induce what we call giant cells in the host plant, and those giant cells at the feeding site sort of continue to provide nutrition to the nematode as it feeds. So once they're inside the root, they become sedentary. They change body shape and have 2 more molts before becoming an adult female. So in the pictures there, you can see the second stage juvenile, which looks just like a worm. Inside the root there, you've got a 3rd or 4th stage juvenile, which is starting to feed and it's started changing shape. And then down the bottom, we've got those pear-shaped mature female root knot nematodes, which they've been obviously excised from roots, those in the photo. Some male nematodes also develop, but they're not essential for reproduction in most root knot species. The length of the life cycle varies with species and temperature. It can be 1 month or less in warm temperatures for some root knot species. So you get several generations in long crops, as I said, which can build up to high populations and a lot of damage. So Guava root knot nematode was first detected in Australia in the Northern Territory in September 2022. It was found in suburbs around Darwin and a nearby horticultural area. And then soon after, in the same year, in December, we found it at a large-scale vegetable farm in North Queensland. That farm in North Queensland had severe root knot infestations, despite having used a pre-plant fumigant and then some additional post-plant nematicide applications. So it was causing really bad damage there. So Biosecurity Queensland initiated a response for 6 months until the end of June 2023. And during that time, we got one further case in a peri-urban backyard, and that was about 2,000 kilometres from the original detection in Queensland. During that time, we processed a lot of samples, more than 150 samples, and then didn't find any more Guava root knot during that time. However, since the biosecurity response finished, we've found Guava root knot at 2 other non-commercial sites in Queensland. So we've now got 4 sites around the state, which are quite widely separated, and we don't really have a good handle on the movement pathways between that site. They haven't found any connections. Like a lot of soil-borne pests and diseases, it's not considered eradicable. So why is this pest a concern? It's highly pathogenic, has a broad host range, and importantly, it has the ability to overcome resistance in a lot of crops. High yield losses have been reported overseas as high as 50 to 70%. And in fact, some sweet potato crops in the US have been total losses because, as I said, it causes direct damage on the produce there as well. So it's a significant threat to Australian plant industries. It was first described in 1983 in China on a tree called the Picara earpod tree. Its name is Enterolobium, so that's where the species name of the nematode comes from. It was very destructive to Brazil's Guava industry during the 2000s, so that's where the common name Guava root knot nematode comes from. So it's had a fairly recent rapid spread around the world, mainly in tropical countries. It may have flown under the radar to some extent because all root knot species look the same, and it's pretty difficult to tell them apart without using molecular techniques. So moving on to some of the research we've done and are doing on Guava root knot. We've got recent or current projects in ginger, sweet potato, and vegetables. The ginger industry was fairly proactive. They were obviously concerned because they've got one of those underground crops that can suffer a lot of damage, and not much was known about Guava root knot on ginger. So in that project, my colleague Dylan, who's online, he validated some molecular diagnostics for our root knot species because it's become much more important to be able to tell which species we're dealing with. Unfortunately, sequencing using some of the common barcoding genes is not a good way to go for. It can't tell apart some of the important root knot species, so we've stuck with species-specific PCR. So Dylan's validated primers for the species we're dealing with, and he's been able to multiplex some of those. We have pure cultures of Guava root knot and other root knot species that we keep in culture. We initiate those from single egg masses from females that we've identified molecularly, so we know that they are pure cultures of one species, and we use those to do crop studies in the glasshouse to look at resistance. and also to identify some resistant rotation crops, cover crops, using those populations. So in our ginger project, in some good news for them, luckily for them, we found that guava root knot was actually not more of a threat than the species that they're already dealing with. That's quite rare for guava root knot. It's generally worse on most crops, but luckily for the ginger industry, we found that it probably will have an equivalent effect to the species they're already dealing with. In the photos there, you can see some lesions on a ginger rhizome and some female nematodes inside a ginger root. However, in sweet potato, as they've found overseas, when we tested some of the popular Australian cultivars, we found that it is worse than the other root knot species. So in the photo on the bottom right there, there are two different sweet potato varieties there. Up the top two pictures, we've got guava root knot. You can see these are only very young spuds, just a short experiment in the glass house, but you can see there's already quite a bit of damage there, even on a root knot resistant variety, compared with the Meloidogyne incognita, the two photos down the right there, down the bottom there, sorry, much less damage during the same time period. We have a national project on guava root knot through Hort Innovation and AUSVEG, guava root knot nematode identification and management. The work areas in that project are improved diagnostics, looking for some high throughput diagnostics there, which will be done by SARDI. We're doing a national root knot survey to update our knowledge on the distribution of root knot species around Australia. That hasn't been done for a while. Uffe Nielsen, from Western Sydney University, will be our collaborator in New South Wales. We'll introduce him at the end. We're looking at some management options for guava root knot, as I said, screening rotation crops, things like that. And there's a communication and extension component, largely done by AUSVEG. We've had some international input at the start of the project from Dr. Johan de Seger, who came across from Florida, who's had a lot of experience with dealing with this pest over the last 10 years or so. So just a bit of information on managing root knot nematodes. Some of the strategies that can be used are on-farm biosecurity, monitoring, using resistant and tolerant cultivars, crop rotation or bare fallows, organic amendments, and chemical control. So I'm preaching to the converted on on-farm biosecurity, but nematodes spread very slowly on their own. It's a figure that's often thrown around is they'll often move about a metre. A patch will spread about a metre in a field in a year if there's no intervention. So human assisted movement is the primary means of dispersal. So that can be through infected planting material or contaminated soil on machinery, boots, etc. So on-farm biosecurity is the best way to keep root knot nematodes and other soil-borne pests and pathogens out of your property. So practicing come clean, go clean, restricting access to areas where people don't need to go, putting up signage. So some of those simple low-cost measures can make a big difference. Monitoring is important for making informed decisions on root knot nematodes. So regular soil sampling and testing, monitoring for symptoms can allow for early detection. We have pre-plant thresholds for many crops, so taking samples prior to planting can help make informed decisions on nematicide use, for example. And it can be important to know what species of root knot you're dealing with, because resistance of rotation crops can vary with the different root knot species. So that also can help to guide management. Resistant cultivars is a popular tactic for dealing with many pests and diseases. As I mentioned, this could be more difficult with guava root knot because it does overcome that resistance in a lot of crops. I gave the example of our sweet potato cultivars earlier being more affected by guava root knot. There's some data there from Dr. de Seger from the US in cucurbits. You can see across the board there, they're pretty much more badly affected by enterolabii, which is guava root knot, compared with Meloidogyne javanica. So research is underway overseas to find and incorporate sources of resistance for guava root knot. Bare fallows can be a good strategy for root knot nematodes, but it must be weed and volunteer free. You can see in the photo there, that's a bare fallow in a sweet potato block, but as you can see, there's a whole lot of volunteers, and each one of those plants is, can support hundreds of thousands of nematodes. So that's just going to carry them over to the next crop. So have to be vigilant with weed and volunteer control for this to be successful. It's not the best strategy for soil health. You can obviously have erosion problems. Excuse me. There's no carbon inputs going into the soil during that time, and you can lose some of your beneficial soil microbes during that period of nothing in the ground. Crop rotation is a good way to manage root knot nematodes. The root knot nematodes are obligate parasites, so the population will drop dramatically if they can't feed on what's growing there. And cover crops obviously provide soil health benefits as well. So we assess cover crops in the glasshouse using our pure populations. And look at the reproduction on those crops. So we try to recommend crops that are resistant or highly resistant, and those categories mean that there's virtually no reproduction taking place on those crops while they're growing. At the moment, we're updating. We have a selection of crops we know are good for root knot nematode, but we're screening those against Guava root knot to see which ones also hold up against that species. Some of the forage sorghums, sun hemp, and some oat varieties are looking promising. Just a note that using cover crop mixtures or biofumigation can be problematic. I know that mixtures are popular, but if 2 out of the 3 things in a mixture are root knot resistant, but one isn't, then it's not really going to do the job. It's just going to carry over the pest. And a lot of the brassicas used for biofumigation are also susceptible to root knot nematodes. So in that case, you're just building up or maintaining the root knot nematode while you're growing your biofumigant, and you've just got to hope that all goes well when you actually incorporate it for the fumigation part of that. Using organic amendments can help to suppress nematodes. High inputs of organic matter improve soil biological health. That carbon from those inputs fuels the food web and increases the diversity and abundance of nematode predators that are naturally in the soil. There are things like nematode trapping fungi, predatory arthropods, predatory nematodes, and parasitic fungi and bacteria. The photo down the bottom there, you can see nematode trapping fungi. It produces traps, in this case lasso type traps, that actually catch the nematodes, and it can use them as a food source. So yeah, things like that are naturally in the soil, and by adding organic amendments, their populations can be increased to suppress nematodes. There's some data there from a sweet potato trial we did where we had a couple of different organic amendments. The most successful was a mixture of sawdust and chicken manure. We also had a compost treatment. The blue bars on the graph there are root knot nematodes. So you can see that it's root knot's been significantly suppressed by those organic matter treatments, and free living nematodes, which are the orange bars, have been boosted. Those samples were taken at harvest, and you can see at that point the root knot had bounced back in the nematicide treatment to pretty high levels. It had provided control early in the crop when it's important, but you can see that the organic matter treatments provided that sort of long-lasting suppression in that trial. Availability and cost of amendments can limit their practicality. There has to be a relatively local source, and can be quite expensive as you need to put on high rates. And you also have to bear in mind that high rates of these organic materials can increase other pests and pathogens. So you just have to be careful. And finally, chemical control, which is not my field at all, but just to cover off on it. As you would know, most of the old school nematicides were pretty toxic chemicals, and most of those are no longer available or are very restricted in their use. Fumigants are still pretty widely used in vegetables, as I understand, and they can be very effective if application conditions are right. But they're obviously not great for soil health if they're killing a broad spectrum of things in the soil. There is a new generation of nematicides available. Most of these are available in Australia now. They're generally less toxic and more specific in their action. Some of them claim to be very specific to plant parasitic nematodes. And some of them are gaining acceptance and are fairly widely used already. But just note that performance and claims of not all of those are fully evaluated yet. So just going back to our national Guava root knot project, the objective 3 of that project is a targeted national root knot nematode survey. As I mentioned, our New South Wales collaborator is Professor Uffe Nielsen, and he would like your samples from New South Wales. So if you have come across any root knot samples, know people that are dealing with it as a problem, please contact Ufa's team. He would love to have them for characterization.